Our second speaker this afternoon is Dr. Wei-Su Hu. Dr. Hu received her PhD in genetics from the University of California, Davis. And then she came to the University of Wisconsin-Madison as a postdoctoral fellow with Dr. Temin. Under Dr. Temin's guidance, Dr. Hu studied the mechanisms of retroviral recombination and reverse transcription. In 1991, Dr. Hu joined the faculty of West Virginia University as an assistant professor in the Department of Microbiology and Immunology, and was promoted to associate professor with tenure in 1998. In 1999, she joined the National Cancer Institute as a senior investigator in the HIV drug resistance program. And she's an American Academy of Microbiology Fellow, the recipient of research awards from the Office of AIDS Research, and a member of the Behavior of HIV Viral Environments Consortium, also known as BeHIVE. Let's welcome Dr. Hu.
Can you hear me okay? Okay, great. Thank you so much for this wonderful occasion. Okay. Thank you so much for this wonderful occasion and this joyful event to celebrate this occasion. I will tell you a little bit about my HIV retrovirus, of course, transfer the genetic information from the RNA in the virus particle to the DNA in the provirus, and then back to the RNA in the virus particle. So they really transfer genetic information in two different forms. And so today I'll tell you a little bit about my personal journey from RNA to DNA with my time in Temin lab. And then I will tell you a little bit about our current work from DNA to RNA, how retrovirus regulate this process. So when for a PhD student, I was studying human alpha-globin genes. And it's a duplicated genes, and if there is a deletion, people suffer a disease called alpha thalassemia. And I was very curious about this because I studied the recombination that make this to happen. So I wonder, we have all these wonderful DNA in the lab, can we put this DNA back to people and alleviate the disease? Now, of course, I wasn't the only one who thought that, but then I thought, how do we do this? And retrovirus sounds like the perfect solution, and because they can integrate the DNA into the chromosome. So then I thought, well, I really don't know how to do this better, so I really better learn more about retrovirus. So I applied for a postdoc fellowship with Dr. Howard Temin, and I was really lucky that I actually got in. But during this time, in his lab, I study how the virus replicate, and one of the things we look at is recombination. So the first question is, when does it happen? It turned out to be retrovirus recombination didn't happen when two viruses infect the same cells, but happen when these doubly infected cells make particles. And some of the particles have a copy of RNA from one parent, another copy from the other parent. And when doing reverse transcription of these particles, recombination can happen. So we figured that, and the second question, how often does it happen? And to do this, we generated two virus constructs, and both encode, they're pretty much the same. They encode a neomycin-resistant gene and a hygromycin-resistant gene. But in one vector, there's an inactivating mutation in neo, and the other an inactivation mutation in hygro. So both parents can only be resistant to one drug. But if recombination happened during reverse transcription, then we could generate a virus that's resistant to both drugs. So to do this, we actually made cell lines that's individually infected cell clones. And then we harvest virus from these doubly infected cell clones and infect new cells. And then the next step we do is put these cells in different drug selection. A single drug selection to look at the parent titer, and the double selection to look at the recombinant titer. So this is done in the late 80s, late 80s. So nowadays we can do a lot of different things, but at that time, this is our tool. What we did is we take the virus, we dilute them down, infect cells, and if you can look at the tray over here in next to Dr. Hammond's hand, and that's a tray of cells with different dilution, infected with different dilution of viruses. And then after the selection is over, we use the microscope here to count the colonies. And that sounds pretty straightforward. So I still remember this really clearly. One day, after months and months of work, getting double infected cells, infect new, and put them in selection, I was about to count colony and find out what the recombination rate is. So I was very excited. And Dr. Temin knows that too. So he said to me, I'm going to Paris tonight. When you find out what the rate is, come to my office and tell me. I said, okay, sure. But then when I start counting, I start asking myself, what should I do? I don't want to bias myself. So what I decided to do is I decided to count them blindly. I decided to count all of the high-growth selection. And by then I wouldn't know what's what titer. Then I'll count all the neo selection. And after that, I'll count all the double selection. But then I couldn't match them. So I was sitting there counting all day, and I noticed that he was kind of dropping by periodically, trying to check. And I was too busy counting, and I couldn't tell him what's going on. And eventually, he couldn't take it anymore. He'd come up to me and say, so what's the rate? I said, I'm sorry, I don't know what the rate is. You see, I just finished counting all the high grow. I'm counting half the neo. I haven't counted double. I have no idea what the rate is. And he looked at me, and I can tell he was a little aspirated. And he's like, okay, why don't you pull up a tray of each match infection and let me count it? I said, sure. So I pull up one infection. He sat down, he started counting. And then he quickly counted several plates, and he looked up to me and he said, 2%. And then he got a big smile on his face, and then he said, call me in Paris when you get the rate. Then I need to show you, this is what we published in the PNAS paper. After I counted all the plates of 10 different infections, it's 1.8%, 2% basically. So my time with Dr. Temin, the recombination study was wonderful. And these studies also have told us a lot about later on application to a recombination. And for Michael discussed that the precursor for HIV-1 is actually a recombinant. And then in addition, many of the HIV circulating strains are recombinants. And recombination can quickly assort mutations in different places, like what Eric talked about. And they can quickly generate drug-resistant variants. And it's been shown that the recombination can do that in HIV. And in addition, they can reassort things and generate HIV strains that's resistant to host immune response as well. So from our drug-resistant study, we can say some of these. But from a personal point of view, Dr. Temin was a wonderful mentor for tell us how to be a nurturing mentor and how to be an innovative scientist. And I have learned to do hypothesis-driven works, and also, as many have mentioned, to be quantitative in your studies. And the other thing is he's always inspired me in his curiosity, his passion to science, and after all, science first attitude. So some personal perspective. So we use reverse transcriptase in our lab every day. So the discovery of reverse transcriptase, of course, changed biology and virology, but also had tremendous impact in biotechnology. But in addition, although I first walked into the lab thinking I want to work on gene therapy, I actually didn't wind up doing that. I was so fascinated by how viruses replicate, I pretty much did all my studies on virus replication mechanisms. However, nowadays, retroviral vectors are used for gene therapy studies, and including those being approved to look at sickle cell anemia and treatment of cancer. And also, I want to share with you that this is many years later. Although we described heterozygous particles, we didn't have physical evidence. But using imaging approach, we actually managed to get evidence that heterozygous particle exists. And this work was done in collaboration with John Coffin and Winnie Pathak, two of the Temin trainees also. And we also later on showed that the recombination is not only just a wonderful way to assort mutation, but it's actually required for efficient HIV reverse transcription. And this work was done by Jonathan Rawson, who was actually got his PhD with Lumansky and did the postdoc in my lab when he did this work. So there are a lot of overlaps in the Temin trainees, and that I'm very proud of. So now I will tell you a little bit about some of our current studies, and that is from DNA to RNA. So HIV-1 and spliced RNA actually have two different functions. The first function is it needs to be translated to generate GAG-PAL to make virus particle. And the second is it's actually packaged into the virus particle as a genome. Both functions are absolutely required to generate infectious viruses. However, how does one RNA balance these two functions, and how do they regulate it? It turned out to be HIV does not make just one RNA. It makes multiple RNAs and splice RNAs using different transcription start sites. So here I'm showing a slew of different RNA, and turned out to be there are two major ones. One of them has 3 guanosine at the beginning that we call 3G RNA. The other has 1 guanosine at the beginning that we call 1G RNA. And these two RNA only differ by 2 nucleotides. So it's being found that although these two RNAs are so similar, they actually are a little different. 1G RNA is selected over 3G RNA to be packaged. So then the question, so when we first heard this observation was first made by Matsuda's group, and then by Alice Telesnitsky and Mike Summers' group. And when we first heard this, we were dumbstruck. We said, how can this happen? So we decided to look at this, and these are our own results. And I can show you that the y-axis is the proportions of different RNAs. You can see the cell, the major one is blue, that's a 3G RNA. In the virus, the major one is red, and it's 1G RNA. So it's very true. So then the real question is, how can 2 9 kb RNA only differ by 2 nucleotides do such different things? So it turned out to be our group, as well as other groups, proposed that what's really different is the 5' UTR untranslated region of these 2 RNA folding to different conformations. And this is done in collaboration with Kareem Muzier-Forsythe, and who's a fantastic RNA structure biologist. And we found that the 1G RNA is folded into an ensemble of different structures. And so is 3G RNA. However, the difference between 2 nucleotides made them fold into different ensembles of structures. So why are these structures make a difference in functions? If you look at here, in the 1G RNA, the signal that's important for RNA dimerization is exposed. In addition, the 2 spots, that's the primary GAG binding sites, also exposed. So this RNA is ready to interact with another RNA to dimerize, and it's ready to bind RNA and be packaged. However, if you look at the 3G RNA, the DIS is actually base paired with somebody else, so it's not exposed. And the 2 GAG binding sites also interact with other sequences and it's not exposed. And therefore, this RNA is actually not ready to package. So then we wonder, why do the HIV actually do this? Obviously, you need the genome, you need one gRNA, but why do you want two? So to address this question, we actually made mutations here so we can make HIV that mostly express 1G or mostly express 3G, but not both. And then we asked, can these viruses replicate? And this is what happened, is we put this assay, we put a little bit of virus in T cells and let them replicate. And we monitor the replication through the particle release. And you can see that all three viruses can replicate. So then the question is, how quickly can they replicate? So this is, you can see that this one is the wild type, and this one is the 3G virus. And this 3G virus actually do not replicate as well because the virus come out at a later time. So we say, okay, 3G virus has a replication defect, a fitness defect. But what about 1G virus? It turned out to be 1G virus actually in this assay looked very similar to wild type. So then we use a more sensitive assay. In this assay, what we did is we take a little bit of 1G virus and some of the wild type virus, and we mix them up at different ratios. And then we put them on T cells and we let them go for a while and say, who wins? And in this, as you can see here, regardless which ratio we mix them in, wild type always wins. In 11 out of 12 experiments, wild type wins. And that tells us that the 1G virus actually has a replication fitness defect also. So these studies tell us that, yes, HIV with just 1G, that express just 1G or express just 3G can replicate in T cells. However, neither virus can replicate as well as wild type, which express both. And therefore, HIV really uses the transcription start site as a regulatory mechanism to optimize their replication fitness. So then our question is, okay, fine. We know why 1G is important. Well, why is 3G important? So one possibility is, what about the other function? Do they translate better? So what we did is we used two different assays. And one is used polysome profiling, and the other is using in vitro translation. And we found that both 1G RNA and 3G RNA are both translated. However, 3G is translated better than 1G RNA. And we also use mutants and show that, again, it's the RNA structure that regulates the translation efficiency. And I want to add that Sydney, in Nate's lab, had very similar result using a different approach, an imaging approach. They also show 3G RNA is translated better than 1G RNA. So from these studies, all these studies are based on in vitro studies and cell culture studies. But what happened to people living with HIV? What happened to the real people? And so what we decided to do is look at patients and see what happened. And for this, we collaborated with another NCI investigator, Frank Maldarelli. And we have identified samples collected from people living with HIV, the same donor at the same day, that we collect two sets of sample. One set is a PBMC, the other is plasma. The PBMC sample, we can look at the cellular expression of HIV. And the plasma, we can isolate virion RNA and ask what's in the virion RNA. So this is what we found in there are three sets of donors. In different donors, this is what happened in the cell. As you can see, what we can tell is HIV, while replicating in human, also use multiple transcription start site. And they also have 3G RNA appear to the most dominant species in the subtype B infected people. And then we look at the virus and we ask what happened to the virus. And turn out to be, in the matched sample, we can say that they are preferentially packaged 1G RNA. So they look very much like what we see in the cell culture. So our cell culture system really actually mimic what's happening in the real infection. So in summary, we found that HIV actually, they use two major transcription start sites to make two RNA. One RNA is really good at packaging, and one RNA is really good at translation. And by expressing these two RNA, they can optimize their replication fitness. And this is also true in not only in cell culture, but also in people living with HIV. So I want to thank a lot of the people in my lab, and I won't read all the names because of the time constraint, but also want to thank some of the my multiple collaborators for all these years. Thank you very much.
Okay, questions for Dr. Hu? We have Nate over there. First question. Can you please wait until the... Thank you.
Yeah, beautiful talk as always. I was always, one of the things that like we've kind of played with in addition to HIV is thinking about, one of our old favorites, murine leukemia virus, and other viruses that don't seem to do transcriptional starts or don't seem to exhibit transcriptional start site heterogeneity. I was wondering what your thoughts are for viruses that don't do it this way in terms of how they're differentially regulating RNA fate.
Yeah, that's a great question. So we have done study looking at multitude of lentiviruses. A lot of lentiviruses do this, but not everybody. And there are some lentivirus that only make one major species. And so we are always very curious about this. And the short answer is I have no idea. And the long form is I think the virus has a way to modulate this. Judith Levin had done this, years 1974, showing that there are two pools of MLV RNA, one for translation, one for packaging. So if it's not transcription initiation, there might be other mechanisms they use to segregate the pools. And I think that's waiting for you to find out what the mechanism is. Okay. Yes, Tom.
Have you thought about or tested whether the decreased translatability of the 1G genomes contributes to favoring reverse transcription upon entry into the cell? As if that is also a functional advantage, because you wouldn't want it to be translated too much.
That's an interesting question. So so one of the former students from John Coffin's lab, has recently shown that some of the retroviral RNA, when they enter cells, can be translated. However, you're going to hear from Renee later that most of the infectious event, the RNA is actually not exposed. And therefore, it's a different story. Yeah.
Any more questions? If not, let's thank Dr. Oh, Nate has one more question.
Yes, please.
Time, right?
Yeah, we have time.
Yeah, another question I've always been fascinated with is there must be a lot of transcriptional start site heterogeneity within the genome naturally. Is there much known about? Are there a lot of cellular transcripts that are regulated this way, or is it something that you guys have looked at? Or?
Yeah, my knowledge of that is reading review articles, but actually HIV is not unique. I think a lot of gene transcript have neighboring sites. That is not unusual. Yes. Yeah, but we can. But as you know, you can also manipulate the HIV sequence to make them mainly start from one. So. How they
just having, two nucleotides are enough to completely change the behavior. You just think of how that might diversify, the genome in additional ways. I was just thinking that's kind of striking.
Right. And the other thing is HIV is incredibly clever And the way they have I have to mention this, the way they have the 3 guanosines, although they package 1 G, oftentimes the G cap is copied. So when they're doing the reverse transcription, having 3G there gives them a buffer zone to protect the genome. So that's part of the mechanism they use.
Okay, let's thank Dr. Hu.
Thank you.